Welcome to the Proteomics in Proximity podcast, where your co-hosts Cindy Lawley and Sarantis Clamidis from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Cindy and Sarantis.
Hello there. Welcome back to Proteomics in Proximity. I'm your host, Cindy Lawley, with my co-host, Sarantis Clamidis. We have a very special episode for you today. We have a few guests coming to us from our new home, our new family, our new neighborhood, Thermo Fisher Scientific. Some of you might recall that last summer we completed the joining of Olink and Thermo Fisher Scientific. I'm excited to report that we've got Gianluca and Karen to join us to talk about why and what that means for all of us and all of you. Sarantis, do you want to tell us a little bit more about our guests?
Thank you. I'm really pleased to welcome our guest today. Actually, really honored to have Gianluca. Gianluca is Executive Vice President at Thermo Fisher Scientific. Karen Nelson, she's Chief Scientific Officer. And we are looking forward to discuss about integration of Olink Proteomics to the Thermo Fisher Scientific family, and not only as a strategic alliance and movement, but also the next step, opening and promoting actually a world that is healthier, that is safer, and actually makes our life easier for everybody. I would like to pass the word now to Gianluca, if you don't mind. I would like to know you a little bit more better and to get to know your journey and your career path that you had in Thermo Fisher. And I'm looking forward to hearing from both of you. Thank you very much.
Sarantis, thank you very much. And Cindy, thanks for having both myself and Karen. We were incredibly excited to have the opportunity to join you. As you might remember, many months back when we first talked, I knew about you, not because of Olink, but because of this podcast that really helped me as a non-scientist over the years to get acquainted with the magic word of proteomics. And having been 20 years in the industry, I always thought that proteomics at a certain point would be very transformative. And indeed, it is the case. And that's why ultimately we decided about a year ago to acquire Olink. And now Olink being part of the Thermo Fisher Scientific family, it is truly being transformative to the world of science. So I'm incredibly excited to be here. A little bit about me.
Can I just pop in and say that Gianluca and I met at the castle in Uppsala at a dinner, and he knew about the podcast, which was just so flattering to us. We were very excited to hear that. Sorry, go ahead, Gianluca.
Totally. I knew about the pod. I was a fan, and I'm continuing to be
You even had it on
fan.
Your phone. You even showed me. I said, "Oh, please, that's..." And he had it up. It was right at the end of the episode. I was gobsmacked.
I had the proof. The proof is in the pudding, you see. It wasn't only marketing.
The first step toward trust between us, Gianluca.
Absolutely. Well, Sarantis asked a little bit about me. I've been at Thermo Fisher Scientific for the best part of the last 20 years. I was in Europe at first, then I spent a few years in South America, in Brazil. I moved then in 2012 to China. Actually, one of the biggest achievements while I was in South America, when I was running what at that time was Life Technologies in South America, is meeting my wife, and we together moved in China in 2012. When we moved in China, we announced that Thermo Fisher was acquiring Life Technologies. We ended up spending 5 years running Thermo Fisher in China, had 2 kids that were born in Shanghai, so I think technically made in China and then imported in the US. When tariff were not a thing back in 2018, we moved to California, and then here in Boston, I now run our product and technology businesses at the company. And as said, I'm incredibly excited about proteomics. We have ample time to talk about that over the course of this podcast. So without further ado, maybe I'll hand it over to Karen to tell us a little bit about herself.
Absolutely. Looking forward Looking forward. Actually, I would like to know. I saw that Karen has a tremendous career, an academic career. She was leading the Craig Venter Institute almost a decade. How was the transition from I mean, it's a common question, right? How was the transition from academy to a big technology industry institution, right? How did you find this? How was your vision when you changed?
Well, Sarantis and Cindy, thanks for having us today. It's such a pleasure to be with you guys. And Cindy, I'm just getting caught up on your podcast, but they really are great. So thanks for doing them.
Aw, thanks.
So I joined 4 years ago. And Sarantis, to your question, probably one of the best career moves of I'm sure it has been the best career decision of my life. I spent close to 23 years in a non-for-profit research world with the Venter team. And they were part of the team that did the human genome. My biggest claim to fame was doing the first human microbiome. So a lot of time spent in genomics technologies. And then by coming over to Thermo Fisher and seeing the breadth and possibilities of what we have, all the way from instruments through consumables in the life sciences, through clinical trials, it has been such an amazing experience for me, both in terms of people and learning and the willingness of everybody wanting to partner. And then, when I heard that you guys were joining the family, it was like, Gianluca and I were like, yes, this is perfect, because it was such a nice complement to what we had already in-house and had the potential to really accelerate what we're doing to make the world healthier and safer. So thanks for having us here today.
Yeah, thanks, Karen, because we feel very welcome.
Wonderful.
I just want to highlight and put on records that she said that was the best move of her life and career. She moved from Craig Venter to me. And so I don't know if I have the same scientific pedigree, I have to say, but I'll take that as a compliment, Karen.
Well, it's been such a pleasure to be here.
He's throwing down the gauntlet, Karen, to Craig.
I know.
I'm thinking maybe we'll hear from Craig.
But just the magnitude and the impact of Thermo Fisher. I am one of those who grew up on Thermo Fisher reagents and instruments and supplies. It was Gibco in every aspect of your lab, for example, and different pieces of equipment and thermocyclers. So it's actually, my opportunity to be back at home, just like you guys are here now.
And I'll say it's not an easy thing to make a transition that layers on what Craig Venter accomplished and what you accomplished with him. I mean, just the whole history. It's just phenomenal.
But Cindy, you're part of that too. You made the jump, so you know what it's like.
Yeah,
right?
But so that's why I'm complimenting us both for having such good
mentors
sense.
And good guidance. Yeah.
That's right. It's all about having a cool technology, having wonderful people around you, and those wonderful people then create a good culture. And I think with that in place, changing the world, feeling like we've got a purpose, I mean, that's just icing on the cake, right? So, Gianluca, we've got mass spectrometry, an amazing capability here at Thermo Fisher. And I will say, as Karen says, I come from the genetics world. So proteins have always been really scary to me. I felt like Olink gives me a little bit of an easing into proteomics. But why Olink in the context of the enormous capabilities at Thermo Fisher using mass spectrometry and the advances that we're seeing in that space?
Yes. It obviously starts from the understanding of the importance of proteins and the importance of proteins to obviously our incredibly complex and fascinating biological systems. And as a leader in proteomics through our mass spectrometry franchise, we had the opportunity to have great insights on what was happening in the market, and also the understanding that not only having solutions that are helping you to go incredibly deep and in a way in an untargeted fashion to study proteins was not enough to be able to provide to our customers the full breadth of what they truly need, which to us is also a high affinity method, high throughput like Olink provides, that truly has been transformational when you look at the result of the UK Biobank in terms of both the amount of information that the UK Biobank cohort has been able to create and the one that is ahead of us, but more importantly, the number of publications and the understanding of correlation between the presence of protein, their level, disease. This has been truly transformational and a pivotal moment, I think, in science, and we'll discover more and more. And so we felt that it was very natural to actually combine the Olink technology with the incredible leadership that we already had in mass spectrometry. And indeed, we are now seeing how engaging with customers, the two technology can be incredibly complementary to have a more comprehensive understanding of the human proteome. And I can't wait to see what's going to happen in the next few years, because I do believe there is going to be transformation in the way that ultimately we actually prevent, then detect and treat diseases that today are not treated as well as they should.
To monitor, right? Who's really at risk? And I joke about the Goldilocks, too hot, too cold, and just right. And I don't want to dismiss your microbiome metabolites, but they are so hot, they move so quickly, right? DNA moves at the pace of generations. That's really slow. It's hard to nudge, but proteins, it's just, it's the baby bear bowl. It's just fabulous.
So Cindy, I wanted to pick up on something you said about proteomics being scary. I don't know if it was scary as much as we didn't have the right tools, right? And now finally we do, right?
Exactly.
And if you think about Astral and Astral Zoom and how deep you can go on a sample now, I mean, it's unbelievable what's changed over the past 15, 20 years in terms of technology development. So we're now in a better comfort space. And to Gianluca's point about integrating the Olink data with the proteomics data, I mean, it's such a tremendous advance for science from where I sit. Yeah.
For sure. And I guess I think of it more as scary from a geneticist point of view, where we had this, you know, certainly in 2007, when we advanced to the massively parallel sequencing and being able to analyze those data, right, that we just shifted the burden from the collection of the data to the reconstituting the data and comparing it to reference genomes. And handling those data, or even transcriptomic data, just feels a little more, it's A, C, T, and G, right? There isn't the conformational or the conformational complexity or the protein-protein interaction complexity. But I think geneticists are really getting comfortable with full-length proteins, and then mass spec is going to be the only way today to dig into proteoforms, to dig into what are the post-translationally modified aspects of those proteins that are actually providing the bulk of those signals. And of course, we're seeing from the UK Biobank data, amazing signatures of 5 to 20 proteins that are predicting disease better than anything a doctor has available today. Like this is, just echoing on what you're saying, John. It's a very exciting time.
I mean, following up on Gianluca's comment about UK Biobank, we see recently also that pharma investing on these data sets, right, of generating real-world evidence, real-world data. And we see that they investing also on platforms like Olink. How would you see, both of you, the role that can play in this platform kind of way of working pharma together with governmental institutions, with academic institutions to enable precision medicine? How do you see Thermo Fisher plays a role there, and having all of this weight and breadth of multi-omics approaches?
It's a great question. And, maybe because I'm the probably only non-scientist of the crew here in the pod.
I disagree.
You disagree?
You're a scientist.
Honorary member.
You are absolutely a scientist.
I learned street fighting, spending a lot of time with customers. But, one thing that I've been thinking a lot, and being at heart a lover of technology and innovation, I've been obviously very impressed with what is happening in the world of automotive technologies and transportation. We now have self-driving cars. I wake up in the morning and my Tesla doesn't even need me to tell where I'm going, because the Tesla knows that more probably I'm going to the office. So I sit on my Tesla and there's already a predefined destination in the morning. And then I put self-driving and the car drives itself to the office. The intervention is very limited. And my hope is that at a certain point, health becomes the same. We are on autopilot. And to be able to do that, you actually need devices and detectors and signals. And I think proteins are providing just that. We actually don't understand everything to the same extent that digital technology is able to understand visual signs and help a car driving itself yet. But I think that's going to be obviously a big transformation. And that is why I think, to your question, pharma companies are so interested now in getting a head start in proteomics, because we've seen, again, through some of the result of the UK Biobank, protein signatures and this polyprotein, you can call it risk scores, in my opinion, but I'll leave it to the scientists on the pod to confirm, is more than risk score. I think very different to look at polyprotein signatures and polygenic risk scores as an example. That's why I think pharma is so interested in this polyprotein signature as an indicator of either an early onset of disease or perhaps an indication of response to therapy. So again, over time, I think the utility of protein signature is going to be quite broad in nature, whether it is prevention, early detection, whether it is response to treatment, and I think more things that can be done through those signatures. So it's quite exciting. Does the interest in the space? The key to me is still generating millions and millions of data points so that ultimately we can apply artificial intelligence and truly get to call it a self-driving. Can I call self-driving human, or is it a bit too provocative? I don't know.
I think what do we want to call that? I mean, it's a really good question. We should come up with a phrase, John. I don't know that it's self-driving human, but it's we're certainly putting power in the hands of the individual, right?
Yes.
We, the community, the scientific community, by building such tools. And I think this point about self-driving, I love the comparison. I love a good analogy. I was talking to I live in the Bay Area. You can't swing a dead cat without hitting somebody who's been involved in some aspect of collecting self-driving. I would never swing a cat, by the way. Is involved in some aspect of this technological advance. And even where I used to live in the agricultural belt of California, they used an old Air Force base to do some of the practicing of that. But what turned out to be the most important thing is the right data were used. The minute you have incorrect data, and here I'm calling back to specificity, right? And the importance of knowing exactly what protein you're measuring. The minute you have wrong data, the algorithm can't discern the difference. And so I think that was the turning point, based upon the conversations I've had with a few of these folks, of being able to really make that work. Because it's phenomenal to see those cars drive themselves. They can come pick you up at the curb with not a driver in the seat. It's impressive.
Actually, we're not far away from the concept of digital twins, right, in pharma company, Gianluca, that they have the digital twins where actually can project things on an AI, right, and predict things on AI. I think we're really close on this. We're really close on this era.
And a virtual cell. Right, we could navigate through
So,
it.
Cindy, just to pick up on one of the points Gianluca was making, is some of the preliminary data coming out of UK Biobank in terms of stratifying patients. It's really amazing, right?
It is.
And so, to Gianluca's point, the early diagnosis, like the protein signature that's coming really early on, we're going to be at a point where we can use that to change the trajectory of health or treatment. And I think we're just at the beginning here, and I just think it's going to change how we think about human health. Could go all the way to animal health, environmental health, but it's just such an amazing opportunity for the field when you start to look at the initial implications of the data that's coming out. And I think, Gianluca, we have multiple collaborations now, right? It's not just UK Biobank. I think we're processing
Top Med, FinGen. Yep.
Yeah.
We're involved in Our Future Health. There's so many places where there's opportunities. And I think, just for context on UK Biobank, what a sweet data set that is, just to remind listeners that, those collections were made for individuals between 40 and 67 years of age. And it's been almost 20 years for them to develop diseases. And so, to Karen's point, to John, to your point, this is a beautiful set to be able to look at those baseline blood samples and see the signatures for the same diseases in different individuals. And that has, certainly coming out of Ruth Travis's group, they've been able to do that in cancer. That hit the UK news last year around how they could see a median of 12 years before diagnosis of cancer. 12 years, that's phenomenal, right? A minimum of 7 years, that was what hit the news. And then, of course, the Carrasco-Zanini paper coming out of Claudia Langenberg's lab, where 67 out of the 200 diseases in just the pilot of the UK Biobank Pharma Proteomics project, they were able to see these 5 to 20 protein signatures that outperform anything a doctor has available today. So just for some context, that's some of the exciting stuff coming out of that.
Yeah.
That study, that pilot. And those pilot data are available for application to use them, and they're free for many, many, many academic institutions. So it's an exciting time.
One thing I want to add, one thing that is maybe the elephant in the room, or the human in the room, call it the way we want.
The self-driving human.
The self-driving human. But in serious terms, one of the things that I've been fascinated with is ultimately human psychology. And I think I'm not misquoting this, but in a study that was run a few years back, they highlighted how 50% of the individuals that should take medicines that are critical due to their chronic condition, and in many cases, life-saving medicines, they're in non-compliance. 50% of them, they're in non-compliance, which tells you the fact that for some reason, humans are not very self-caring, if you wish. And so one of the things that I think is going to be very important, to your point on the amount of available information, is how do we disseminate this information? How do we make sure that there's enough learning opportunities, both for doctors, but also for individuals, to become more self-aware on how they actually can stop the car before the car crashes? With the analogy of self-driving cars, a self-driving car normally doesn't crash. And I think from a health standpoint, we should make sure that we prevent disease more effectively going forward with all of this insight and information that we have available. But one of the concerns I have, I don't know your perspective, is how do we influence culture? And who is going to be actually helping consumers to do a better job in taking good care of themselves? That to me is still an unresolved issue. If we don't resolve it as a society, then it will be very difficult to take full advantage of these tools. But maybe there's alternative solutions that will be implemented over time as a result of now much better tools to help consumers and patients.
Karen, do you have ideas about that from the genetic space, right? This feels like a parallel of education needed.
Right.
What helped us there from your perspective?
Well, I mean, to Gianluca's point, it has to happen in parallel, right? To get absorption into the general population. I think back then we had set up aside funds for like ethical, legal, and social implications. Remember all of that good stuff, Cindy? And it was a percentage of all funding going into genomic research was used for like education and legal work and getting Because there were a lot of questions, right? A lot of concerns, and not everybody is trained in science, and so they don't understand. We tend to be positive and can only try to discuss the positive implications, but there are concerns about some of these research areas that people don't quite understand. And I think the education and the value on highlighting, I like the car analogy, Gianluca. You're saying that when the light comes on the car, take it in and don't wait till the engine shuts down, right? And we're getting to the point where we can have that light come on really early when there's a problem in the car. Not on an Italian car, though, right? I'm talking about in the other part of the world. So, but I think as a society, Cindy, we probably need to start having those conversations about getting the information out, getting the positive findings out. It's right now it's so positive. How do we get that out in words that the general community can understand? And I think it's up to us to try and accelerate that.
And isn't it easier because proteins aren't tied to our personal genetic information, right? Don't we think that might be because, it's not an identifier of us as individuals, our protein signature, right?
Right, but it's hard to understand, right? Because you talk about proteins and proteoforms and splicing and all this good stuff. And so it's about how to translate that in words that the average person can understand the benefit of the research that's ongoing right now.
Right.
Yeah, I think to your point, it's also really important not only to generate the data, right, but having the tools to interpret the data and make sense out of this data. And I think that's also really important. It will come to the future, and it will come really important to the future as well. Interpret the data and make them easy for the people to understand and make the right use on them. Yeah, that's actually the point, Karen. Actually, I would like to follow up on this, Karen. You have this NGS background and experience. What excites you more in this NGS proteomics nowadays? And do you see this like a game changer for clinical trials, for example? Do you see that will make clinical trials smarter, having an NGS proteomics integrated to the omics pipelines, for example?
Quick answer is yes. You think about NGS, it's like a static point in time that you're understanding what's going on in the cell. Now we can actually understand over a time period how a cell is behaving, how it's reacting, what it's doing. You can do it with a human, you can do it with a bacterial cell, you can do it with a plant. You can understand what the DNA has resulted in terms of activity at the cell level. And I think it's so exciting, right? Genomics was exciting. We built massive informatics resources, software resources, massive tools, AI. We were using all these tools back then. But now we have a chance to actually understand what are the messages coming out and what are they turning into, and not that we want to stop it, but how can we use that information to influence the outcomes and our health in the long term? And I think it's going to, over time, become a critical part of a companion part of clinical trials. I would think so. I don't do that on a daily basis, but I would absolutely believe that you can take a plasma sample on a daily basis and look at how certain protein markers are fluctuating. Imagine a time like that, right?
Well, Chris Whelan's talked about this in depression, where they had 3 endotypes based on proteins after the clinical trial, right? So this isn't in that trial, but they responded differently to the therapy.
Right.
Each of those 3. Yeah. Sorry. Go ahead, Karen.
No, I was just saying that I think you guys had a partnership where initial outputs from Olink data turned actually into diagnostic in the clinic, right? And it took a couple of years, but I believe that that's going to become at scale, that we can have these early biomarkers being detected as a result of the technologies that we're fostering in-house. I really believe that.
Yeah. Yeah, those application and disease-specific signatures that our customers are building. I think that is one of the most exciting things I was excited about in joining the Thermo Fisher family, is that we can accelerate those needs, right?
And Sarantis, my last happy point. Imagine being able to live through the genomics revolution and landing where I can live through the proteomics revolution.
That's amazing.
Not a lot of people get that opportunity, right, Cindy?
So That's right.
Perfect.
That's right. It's pretty special.
Yeah.
It really is.
Would you think also this will open the door to, let's say, FDA and to other, institutions that can be biomarkers that are approved? Will make probably all of this procedure much easier, having all of these nice data sets? What is your opinion, Gianluca, on that?
Yeah, I think regulators have their hands full to stay up to speed with technology for sure. And it's actually refreshing to see that I think we had recently one of the first AI-driven biomarkers approved in the pathology space, if I'm not mistaken.
Yes.
And so it tells you that they're starting to move at pace, and that's a very good news. By the way, I ChatGPT the data point that I quoted on non-adherence, and in fact, there was a study from the WHO that says that non-adherence can account for up to 50% of treatment failure. And just to put this in context, in the US, around 125,000 deaths and out to 25% hospitalization each year that are driven from non-adherence with medication. So it's indeed highlighting how the need of actually information and education of the patient population is super critical. But also I think technology plays a big role. Right? We're seeing
Sentry mode, right, Gianluca? I mean,
sentry mode, yes, and
Get
and
a
also
pop-up.
it's gonna pop up. But in that case, you obviously have the car that allows you to do that. In patient world, actually, you need technology to be able to detect, as Karen was highlighting, what about detecting this biomarker on a continuous basis?
Yes.
And I feel we're still in the era of mainframe in the life science space. Like,
Yeah.
you have this big
Or it's the
instrument sitting in a
Volkswagen
lab.
Bub from 1966.
Yes. That's a good analysis. I'm Italian,
Beautiful
so
car, but not
we
as
use Fiat.
sophisticated.
Yeah. I know that
You
in
know
the
you're
in
in a
the
Volkswagen.
US, you say Fiat means fix it again, Tony. But that's, as an Italian, I could take offense for that. But actually,
Now
it's
you'll
very
get in trouble.
was my first car, and I truly enjoyed my Fiat. But I think technology will need to play a big role, and I could envision over time we already have much smaller and compact sequencers, and as we know, the detection technology for Olink is sequencing, but over time we're going to have smaller and smaller technologies that can be ultimately integrated in our houses and will probably benefit from an availability of biomarkers at a level. And to a certain extent, even with a WHOOP today, you can have these digital biomarkers that are augmenting your insights on your health. And I think this will happen over time for things like protein, for sure, which are challenging to measure, as you know, with the current technology, but they're becoming easier and easier to measure with modern technologies.
And then you think that, let's say, one of the barriers you have to go is like finding new ways of sequencing or more easy ways of sequencing or more handy ways of sequencing, right? That will be the future, how you envision it.
Totally. That's why we're investing $1.3 billion, $1.4 billion every year in R&D. That's Karen's word. She's very, very tightly managing the priority around everything that we spend in innovation. But certainly that's the direction we're driving towards. And I'm sure Karen has some perspective on that.
No, I agree. I was laughing to myself because I remember in the early days of the microbiome, they wanted to put a little detector in the toilet. And so people could know if they were sick. But think about it, right, Cindy? I mean, imagine being able to look at inflammation in a urine sample. It could become our future that, you have some way to check at home, and this becomes a part of your lifestyle. So I do agree with that 100%. But in terms of our investment in R&D and pushing the envelope and integrating cross-company, these opportunities, again, it's the things that wake you up in the morning excited to go to work. We think about these opportunities. And I will tell you honestly that Gianluca and I think the proteomic space is one of the, especially bringing Olink in, is one of the biggest, most exciting areas for us. So we don't advertise it all the time, but we are very proud of the acquisition and what it means for the company. So,
I love to hear it.
very welcome on board. Very happy to have you on board.
Thank you.
Thank
It's been a year now, Sarantis. It's like I've known you forever.
Yeah.
So, but it's great to have you.
No,
Yeah, we get under your skin. We get in
we
there.
are looking forward to
It's very exciting.
It's very exciting.
Yeah.
And it's very exciting to see that the matching, right, with the omics pipeline that you're
Correct.
having, right? And it's really clear, nice fit on that, because now it's a complete toolbox, right, end to end for the customer, from data generation also to data interpretation. That's the beauty. That's the beauty.
Yes, correct.
Can I add one thing?
Oh,
Yeah,
yes,
this is-
please.
I want to
please.
add one thing which is super important, because obviously the Olink technology, as well as largely mass spec for untargeted protein detection and quantification, has been used in the translational and research space. But one of the big unlock will be when those technologies are getting to the clinics. And I think we have an interesting parallel with what we did with Ion Torrent. We acquired the company back in 2011, I believe. And now we are basically the enabler of a vast majority of clinical testing that are next generation sequencing based, thanks to the fact that the team has worked to streamline that technology. Now we have a fully integrated and automated NGS that is used for therapy selection with great products. We just announced that we got FDA approval for our Genexus system and a set of panel and markers that are used for therapy selection. So it's incredibly exciting. It's been a long journey, but it's a journey that we learned how to go through and execute. And I do envision the same journey on a shorter timeline, because we learned a lot over the last decade, 15 years. And I'm super excited thinking that when we're going to see proteomics into the clinics at scale. It will require obviously content, technology, etc. But it's exciting to think of when it's going to happen.
And I think what's critical, and we do this today with our focus panels, is being able to turn over those custom subsets of proteins that we already understand exactly how they cross-react together. We already understand how they play, and those, authentication steps of, 2 antibodies and 1 hybridization, that 3-factor authentication is really enabling this to happen quickly. And so I would encourage folks to keep an eye on this space, because we're already seeing, there's over 700 references to the UK Biobank, foundation paper that came from the pharma partners. That was in October of 2023. So lots of references by using those data, some of them just mining the data for new insights.
Yeah.
It's a rich source.
And if you don't trust a 1-factor authentication for your digital needs, why should you trust it for your health?
That's true.
So
Exactly.
Use Olink. That's the only 2-factor authentication at scale out there. So.
And antibodies are what we use in therapies, right? So I'm also excited, although much of it's behind closed doors, but when pharma are able to publish on mechanistic insights from the therapeutic targets that they're identifying, right? This is a big push for why 14 pharma partners are paying for 600,000 samples to be run, the entire cohort in the UK Biobank. And I think that the mapping of those causal pathways across different diseases is going to be a reference that we will come back to for many years. It feels
over and over again.
Karen, it feels like the GWAS revolution over again, right? It, like you say, it's just such a privilege to have a front row seat and a tiny impact on where this goes and where it will go next. So thank you for having us.
To be part of the team, right, Cindy? I mean, we're all part of the team, right? So
it's awesome. It's awesome. Yeah. So I'm having the time of my life. I am having the time of my life. How about you, Sarantis?
That's great. No, that's great. I mean, I'm coming from the omics perspective, right? And being in a big family with the omics capabilities coming together, for me, is the most exciting thing ever. Looking forward to bringing to clinics, looking forward to bringing biomarkers and push and convince FDA to have biomarkers for clinical diagnostics. That's I think the next step we'll have to go through.
And Cindy, one more plug. One more little comment. Tim Miller, who is on our team, sent me a list of about 10 microbiome papers where they're using Olink to look at inflammation and doing that correlation. So
thanks for the reminder.
But it's just amazing how it's spanning multiple different areas of science, right? That you probably wouldn't have thought about the connection originally.
Absolutely.
That's right. And we can put those in the show notes. So if folks are interested in that, I had just had a customer ask me specifically about that as well. Another person who came from that microbiome. Oh, it's still happening, this microbiome.
Still trying.
Revolution, where we're learning so much. Yeah, it's like I always think of them paying, the biome is paying currency, like it's paying rent to live in our body, right? By giving us these metabolites like serotonin and things like this. Again, Gianluca and I think both really love a good analogy. So final thoughts. We're just going to kind of wind down here. It's a pretty great place to end on. Gianluca, do you have some final thoughts you'd like to leave our listeners with?
Absolutely. The future is bright, and is bright thanks to for us, 120,000+ colleagues at the company showing up every day, trying to create those incredible innovations that equally talented and incredibly important customers and partners are using to actually transform the way that we identify early, hopefully, then ultimately identify treatment and treat diseases that today are either not well treated or untreatable. And that's why it is incredibly exciting. To do that well, you actually need the right technology. Back to the analogy of the self-driving car, you need the right sensors, you need the right orchestration information, and you want to rely on something for your life when you drive on a self-driving car, or when you actually drive through your life and you make medical decision. You actually want to rely on the best of the technologies. And our two-factor authentication at Olink is something very special. And I think it has been proven, testified by the adoption that we've seen from the UK Biobank study as an example, and the many partners in the pharma space that now are working with us to create the next generation of medicine and diagnostic. So it is incredibly exciting. It has been a great journey thus far, and I can't wait for the next decade to play out so that together as an industry, we actually can truly transform the way that medicine works.
Beautiful. Karen.
Cindy, I can't add anything more to that. I'm good. Gianluca, that was perfect. That was really good. So thank you again for having us. It's been really
absolutely
a lot of fun.
So looking forward to it. So in the show notes, I'll add some of the genetic corroboration of the Olink specificity and also some mass spec corroboration of the Olink specificity, as well as ELISA, right? There's lots of ways to corroborate. You just have to be able to see the protein in whatever matrix you're looking at. Any final words from you, Serantis?
No, I mean, it's exciting times, and looking forward to break the barrier, right? To make our world healthier, safer, right? And the best place to live. Thank you very much, for your time, and it was a great honor to have you, both of you, here.
Thank you both.
Awesome. Well, just as a reminder, if you want to reach out to us, we're at pip@olink.com. That stands for Proteomics in Proximity. We love to hear from you. We love to get feedback and questions that you'd like to hear answered here. Thank you very much for tuning in. Well, that wraps up this episode of Proteomics in Proximity. Huge thanks to our guests and authors of such impactful publications. I also want to thank you for tuning in. Really appreciate you being here. If you enjoyed the content of this episode, please think about sharing it with friends or colleagues you think might be interested in the content. In addition, if you'd be willing to head over to Apple or Spotify or wherever you digest your podcast and give us a rating and review, this will help others find the podcast when they're searching for proteomics or precision medicine podcasts. And mostly, I want to say we would love to hear from you. So we have a dedicated email address, pip@olink.com. Please reach out. Let us know what you're interested in hearing about, what you care about, and any feedback on the episodes that we have already done so far. This is all about you, and so we're really keen to make sure that we're meeting what you'd like to hear about. Thank you so much, and we'll see you soon.